2 miles beneath the surface, in darkness that has never known sunlight, water erupts at 750 degrees Fahrenheit. It should be steam. It should be impossible. But the pressure down here is so immense that the water stays liquid, superheated, loaded with minerals and gases from Earth's molten core. And in this boiling, toxic, lightless hell, life doesn't just survive. It thrives. Giant tube worms 8 feet long, with no mouth, no gut, no way to eat. Yeti crabs farming bacteria on their own claws, microbes that breathe sulfur and exhale energy, running on chemistry we thought was impossible. They don't depend on the sun. They never have. They answer to a different star, the molten heart of the planet itself. We've been looking for alien life in the wrong place. We've aimed our telescopes at distant moons and frozen oceans light years away, searching for biology that looks like ours. But here, on Earth, in the deepest trenches of the ocean, there's an entire world that evolves separately, that follows different rules, that might be the closest thing to extraterrestrial life we've ever encountered. And it's been here all along. What if the origin of life wasn't a warm pond touched by lightning? What if it began in fire and darkness, in scalding water erupting from cracks in the ocean floor? What if the chemistry of life is stranger, older, and more alien than we ever imagined? Tonight, we're going where light has never reached, where the rules of biology were written differently, where something that might as well be alien is still evolving in total isolation 2 miles down. This is Omni. Stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture the bottom of the ocean, not the shallows where divers explore coral reefs and sunlight filters through blue water. Not even the continental shelf, where trawlers drag nets and submarines map the terrain. Go deeper, past the last rays of light, past the zone where anything green can grow, down through 2 miles of water pressing down with the weight of 1,500 atmospheres. Pressure that would crush a human body instantly, that would collapse a submarine like a tin can. Down here it's cold, near freezing. The water is still, silent, black as the space between stars. Nothing moves, nothing grows, or so we thought. Then, in 1977, a deep-sea submersible named Alvin descended into the Galapagos Rift, a crack in the ocean floor where 2 tectonic plates were slowly pulling apart. The scientists on board were geologists, looking for evidence of volcanic activity. They weren't expecting life. There wasn't supposed to be life. But as their lights swept across the Rift Valley floor, they saw something impossible. Clouds of shimmering water, rising from the seafloor like smoke. Not cold, but hot, superheated, mineral-rich, billowing up from cracks in the rock, and surrounding these vents, packed shoulder to shoulder, were creatures. Huge white tube worms, their red plumes waving in the current. Clusters of mussels and clams. Crabs and shrimp, pale and ghostly, scuttling across the rocks. An entire ecosystem thriving in the most hostile environment on Earth. In total darkness. In scalding, toxic water. 2 miles from the nearest photosynthesis. It shouldn't have been there. And yet there it was. The scientists stared through the portholes of Alvin, speechless. One of them would later say it felt like discovering life on another planet. Because in a very real sense, that's what they'd done. They'd found a biosphere that didn't run on sunlight, that had never run on sunlight, that operated on a completely different energy source, using chemistry we didn't know was possible. We've spent billions of dollars searching for life beyond Earth. We've sent probes to Mars, analysed the atmospheres of distant exoplanets, planned missions to Europa and Enceladus. Ocean worlds buried under miles of ice, where liquid water might harbour microbial life in the darkness beneath frozen crusts. But we already have that world. It's here, at the bottom of our own ocean, and the life we found there might hold the answer to the oldest question in biology: how did life begin? Because if life can emerge from superheated water, from toxic chemicals erupting from the Earth's core, from darkness and pressure and conditions we considered uninhabitable, then life might be far more common in the universe than we ever imagined, and far stranger. Tonight, we're descending into that alien world. We're going to trace the chemistry that powers these ecosystems, the evolutionary adaptations that allow creatures to survive where nothing should, and the theories that suggest this, not a sunlit pond, not a shallow tidal pool, might be where life on Earth actually began. We're going to explore what happens when biology abandons the sun and learns to harvest energy from the planet's molten core, when organisms evolve in complete isolation for hundreds of millions of years, when chemistry becomes life in ways we're still struggling to understand. And we're going to ask, if this is what life looks like when it evolves independently on the same planet, what might it look like when it evolves on a completely different one? Let's begin where all Genesis begins, in the dark, in the heat, in the chemistry that shouldn't work but does. Maybe we've been defining life by the wrong star. Let's go back to February 1977, the research vessel Knorr, floating somewhere in the eastern Pacific, 200 miles north of the Galapagos Islands. On board, a team of geologists preparing to send the deep-sea submersible Alvin down to the ocean floor nearly 8,000 feet below. Their mission wasn't biological. They were hunting for evidence of seafloor spreading, looking for hot springs that theory predicted should exist where tectonic plates pulled apart. The scientists, Jack Corliss, Jerry Van Andel and John Edmund among them, expected rocks, maybe some interesting mineral deposits. What they found changed everything we thought we knew about the requirements for life. Alvin's descent took over 2 hours. The 3 people crammed inside the titanium sphere watched through small portholes as sunlight faded to blue, then to black. The pressure gauge climbed steadily. By the time they reached the bottom, the ocean above them weighed as much as a freight train balanced on every square inch of the hull. Their light swept across a barren volcanic landscape. Dark basalt, fractured and lifeless, stretching in every direction. Then the pilot spotted something strange. The water itself looked wrong, shimmering, distorted, like heat haze rising from asphalt in summer, except this was 2 miles underwater, where everything should be uniformly cold. They followed the shimmer, and then they saw the vents, chimneys of mineral deposits, some as tall as buildings, others just cracks in the rock, all of them belching superheated water into the surrounding ocean. The water was clouded with dissolved minerals, iron, sulfur, manganese, copper, precipitating out as it mixed with the near-freezing seawater, creating those shimmering plumes they'd spotted. But that wasn't the impossible part. The impossible part was everything else. Clustered around the vents, packed so densely they seemed to be growing on top of each other, were organisms. Huge white tubes, each one 6 to 8 feet long, rooted to the rock, swaying slightly in the current. Blood-red plumes extended from their tops, waving like feathers. Thousands of them. As the submersible's lights played across the scene, the scientists realized they were looking at an oasis, a garden, in the most inhospitable place on Earth. According to Jack Corliss, who was in Alvin that day, the first words spoken were something like, "Isn't the deep ocean supposed to be a desert? The textbook said so. The ecological model said so. Without sunlight, without photosynthesis, there couldn't be complex life. There might be a few scavengers, subsisting on organic matter that drifted down from the surface like snow, but not this, not abundance." They moved closer. The tube worms weren't alone. There were mussels, some as big as a human fist, their shells tightly closed, white crabs, eyeless, crawling across the rocks, clusters of pale shrimp, anemones, fish. An entire ecosystem, layered and complex, all of it concentrated within a few meters of the vents. The scientist knew immediately they were seeing something no one had predicted. This wasn't a handful of survivors clinging to existence. This was a thriving community. And the energy source wasn't the sun, it was the Earth itself. Over the following months and years, as more submersibles descended to vent sites around the world, the picture became clearer and stranger. These weren't isolated anomalies. Hydrothermal vents dotted the ocean floor wherever tectonic plates diverged or converged, along mid-ocean ridges, near volcanic arcs, in back-arc basins. Each one hosted its own ecosystem, sometimes with species found nowhere else on Earth, evolving in isolation, cut off from the surface world by 2 miles of water and millions of years. But how? How could life exist here? The answer lay in the chemistry of the vents themselves. To understand that, we need to understand what's actually happening when a hydrothermal vent erupts. Deep beneath the ocean floor, the Earth's mantle churns at temperatures exceeding 2,000 degrees Fahrenheit. Where tectonic plates pull apart, this molten rock rises, cracking the crust, creating pathways for seawater to seep down into the fractured rock. The water descends, getting hotter with every meter, reaching temperatures of 700 to 750 degrees Fahrenheit. At these temperatures, at the surface, water would instantly boil. But down here, under the crushing pressure of the ocean above, it stays liquid. This superheated water isn't pure anymore. As it travels through the rock, it dissolves minerals: iron, zinc, copper, sulfur, hydrogen. It becomes a chemical soup, loaded with compounds that would be toxic to most surface life. Then, heated and pressurized, it erupts back up through cracks in the seafloor, billowing into the near-freezing ocean like smoke from a chimney. The contrast is violent. Superheated, mineral-rich vent fluid mixing with water that's just above freezing. The dissolved minerals precipitate out instantly, forming those chimney structures, some black, some white, depending on the mineral composition. Black smokers, rich in iron sulfide, can grow several inches per day, building towers 30 feet tall in a matter of years. White smokers, cooler and rich in barium and calcium, grow more slowly, creating delicate formations that look almost crystalline. But here's what matters for biology. That chemical soup isn't just toxic, it's fuel. Hydrogen sulfide, one of the primary chemicals in vent fluid, is poison to most life. It disrupts cellular respiration, binding to the same enzymes that oxygen uses. Inhale enough of it, and you'd suffocate even in a room full of air. But there are microbes, bacteria and archaea, that don't just tolerate hydrogen sulfide, they eat it. These microbes have enzymes that can pull electrons off hydrogen sulfide molecules, using those electrons to power the synthesis of organic compounds from carbon dioxide. It's a form of metabolism called chemosynthesis, and it works on the same fundamental principle as photosynthesis: harvest energy, build sugars, but the energy source isn't light, it's chemistry. Research published in Science in 2016 by a team at the Woods Hole Oceanographic Institution confirmed that these vent microbes are the foundation of the entire ecosystem. They're the primary producers, the phytoplankton of the abyss. Except instead of using chlorophyll to capture photons, they use enzymes to capture electrons from sulfur compounds. The tube worms, those impossible 8-foot organisms with no mouth and no digestive system, survive because they're full of these bacteria. Billions of them, living inside specialized organs called trophosome that fill most of the worm's body cavity. The worm has no way to eat. Instead, it extends that red plume into the water, and the plume is packed with hemoglobin, the same molecule that carries oxygen in your blood, but modified. This hemoglobin binds both oxygen and hydrogen sulfide, two chemicals that normally destroy each other, and transports them safely to the bacteria inside. The bacteria use the hydrogen sulfide as fuel and the oxygen to run the reaction. They produce organic compounds, sugars, amino acids, fats. The worm absorbs those compounds, using them for energy and growth. It's a perfect symbiosis. The worm provides the bacteria with a safe environment and a steady supply of chemicals. The bacteria provide the worm with all the nutrition it needs. Neither could survive without the other, and this isn't a unique case. The yeti crab, Kiwa hirsuta, discovered at vents near Easter Island in 2005, has dense mats of bacteria growing on the bristles that cover its claws. According to research from the Monterey Bay Aquarium Research Institute, the crab waves its claws through the vent fluid, cultivating the bacteria, then harvests them by scraping the bristles across its mouth. It's farming. Bacterial agriculture, 2 miles underwater, in the dark. The mussels around the vents, also full of symbiotic bacteria. The shrimp, they have bacteria living in their gill chambers. Even some of the fish have bacterial partners. The entire ecosystem is built on this relationship between animals and microbes, and the microbes are doing something we thought required sunlight. They're making food from nothing, from poison and heat and rock. This is why the 1977 discovery wasn't just about finding life in a new place. It was about discovering a second genesis, a second way for life to bootstrap itself into existence. For billions of years, we thought photosynthesis was the only game in town. That all complex life ultimately depended on green things turning sunlight into sugar. But here was proof that life could build entire ecosystems on a completely different foundation. And if it happened here, on Earth, hidden at the bottom of the ocean for all of human history until 47 years ago, where else might it be happening? On worlds we've dismissed as lifeless because they're too far from their star, too cold, too dark. What if we've been looking for life in all the wrong places simply because we defined it by the star we happen to orbit? Let's talk about what shouldn't be possible. Every biology textbook for decades started with the same fundamental principle. Life on Earth runs on the sun. Plants capture photons, convert them to chemical energy, store that energy in the bonds of sugar molecules. Herbivores eat the plants, carnivores eat the herbivores, decomposers break everything down, but trace any food chain back far enough and you arrive at photosynthesis, at chlorophyll and sunlight, at the ultimate energy source for every living thing on the planet. Except that's not true. And it never was. The microbes at hydrothermal vents don't photosynthesize. They can't. There's no light. But they're still primary producers, still building organic molecules from inorganic raw materials, still supporting entire food webs. They're just using a different equation. The chemistry is called chemosynthesis, and it's older than photosynthesis by potentially billions of years. Here's how it works. Photosynthesis, simplified, looks like this. Carbon dioxide plus water plus light energy equals glucose plus oxygen. The plant takes CO2 from the air, splits water molecules to get hydrogen, and uses energy from sunlight to forge those ingredients into sugar. Oxygen is the waste product, exhaled into the atmosphere. Chemosynthesis at a hydrothermal vent runs on a parallel logic but with completely different inputs. Carbon dioxide plus hydrogen sulfide plus oxygen equals organic matter plus sulfate plus water. The microbe takes CO2 dissolved in seawater, strips electrons off hydrogen sulfide, and uses the energy from the oxidation reaction to build sugars and other organic compounds. Sulfate, basically sulfur bound to oxygen, is the waste product, released back into the water. No light, no chlorophyll, just chemistry. The key is the hydrogen sulfide. When seawater circulates down through cracks in the ocean floor, heated by proximity to magma, it leaches sulfur compounds out of the basalt rock. That sulfur gets reduced, gains electrons, becoming hydrogen sulfide, H2S. This is the same chemical that gives rotten eggs their smell, that's used as a chemical weapon in high concentrations, that binds to the enzymes in your cells and shuts down respiration. But to certain bacteria, hydrogen sulfide is fuel. It's energy dense. When you oxidize it, strip those electrons back off, you get a surge of energy. Not as much energy as burning glucose with oxygen, but enough. Enough to power the construction of complex organic molecules, enough to sustain life. The bacteria doing this are called chemolithotrophs, from the Greek for rock eaters. They're harvesting energy from stone, essentially, from minerals dissolved in water, and the diversity of strategies they use is staggering. Some species oxidize hydrogen sulfide, others use hydrogen gas, H2, which also bubbles up from the vents. Still others oxidize methane, or iron, or manganese, or ammonia. According to research published in Nature in 2019 by microbiologist Victoria Orphan at Caltech, there are entire guilds of microbes at hydrothermal vents working in metabolic partnerships, each species performing one step in a complex chemical relay, passing electrons and molecules to the next organism in the chain. One bacterium might reduce sulfate to sulfide. Another oxidizes the sulfide back to sulfate. They're cycling the same sulfur atoms back and forth, over and over, and at each step energy is extracted. It's like a chemical perpetual motion machine, except it's not perpetual. The energy ultimately comes from the heat of the Earth's interior, from the geological processes that keep the vents flowing. And this works. It works so well that these bacterial mats can achieve population densities that rival the most productive ecosystems on the surface. A single square meter of microbial mat around a hydrothermal vent can contain trillions of cells, all chemosynthesizing, all building organic matter from dissolved gases and minerals. The implications are profound. For one thing, Chemosynthesis doesn't require any of the conditions we thought were essential for primary production. It doesn't need light, so depth doesn't matter. It doesn't need oxygen. Many of these bacteria are anaerobic, meaning they evolved in environments where oxygen was absent or even toxic. It doesn't need moderate temperatures. Thermophilic bacteria thrive at temperatures that would denature most enzymes, some growing happily at 250 degrees Fahrenheit. This means the habitable zone for life, the range of conditions where life can exist, is far broader than we realised. You don't need a planet in the Goldilocks zone of its star, where liquid water exists on the surface under a stable atmosphere. You just need liquid water, some minerals, and a heat source. Any heat source. Which brings us to the question of origins. If chemosynthesis is older than photosynthesis, and there's strong evidence it is, then life might not have begun in a warm sunlit pool sparked by lightning, slowly assembling amino acids and nucleotides until something self-replicating emerged. Life might have begun in the dark, in scalding water, at a hydrothermal vent. This theory, called the hydrothermal origin hypothesis, has been gaining traction since the 1980s. The argument goes like this. Early Earth, 4 billion years ago, was a violent place. The atmosphere was thin, dominated by carbon dioxide, methane, ammonia, no oxygen, no ozone layer to block ultraviolet radiation. The surface was getting pummeled by asteroids and comets. Oceans existed, but they were hot, acidic, and probably shallow in many places, frequently vaporized by impacts. Not a great place for delicate organic chemistry to slowly build complexity. But deep underwater, insulated from the chaos above, hydrothermal vents would have been stable, protected, and they would have been providing exactly the ingredients life needs: heat, minerals, chemical gradients. The interface between hot vent fluid and cold seawater creates natural chambers in the rock, tiny pores where molecules can concentrate, where reactions can proceed in an enclosed space rather than dissipating into the open ocean. Research by geochemist Michael Russell and his team, published in multiple papers over the past 2 decades, has focused on a specific type of vent called an alkaline hydrothermal vent. Unlike the black smokers, which are hot and acidic, alkaline vents are cooler, around 100 to 200 degrees Fahrenheit, and produce fluid that's basic, rich in hydrogen and methane. These vents create structures called chemical gardens, delicate mineral towers riddled with microscopic pores. Russell's hypothesis is that these pores acted as natural reaction chambers, protocells where the first biochemistry occurred. The mineral walls of the pores contain iron and nickel compounds, the same metals that sit at the active sites of many enzymes today. The pores themselves create a pH gradient, acidic ocean water on one side, alkaline vent fluid on the other, and that gradient can be used to drive chemical reactions, the same way a battery uses a voltage difference to push electrons through a circuit. In 2019, a team at University College London demonstrated in the lab that simple organic molecules, fatty acids and amino acids, can spontaneously form vesicles and proto-metabolic cycles under simulated alkaline vent conditions. No enzymes, no DNA, just chemistry, proceeding naturally when the right ingredients and energy gradients are present. The chemicals organize themselves. This is radically different from the classic primordial soup model, where life is supposed to have arisen from a dilute broth of organic molecules floating in the open ocean, randomly bumping into each other until something clicked. The hydrothermal model says life didn't arise from randomness. It arose from structure, from enclosed spaces where reactions could be concentrated and sustained, from energy gradients that could be harnessed. And the metabolism came first, not replication, not even information storage, just the ability to harvest energy from the environment and use it to build more complex molecules. Chemosynthesis before genetics, chemistry before biology. If that's true, then the first organisms weren't free-floating. They were anchored, growing in and around hydrothermal vents, extracting energy from hydrogen and sulfur, slowly assembling the machinery of metabolism. DNA came later. Membranes came later. Photosynthesis came much later, after some lineage of chemosynthetic bacteria figured out how to strip electrons off water instead of hydrogen sulfide, unlocking the vastly more abundant energy source of sunlight. But the root, the base of the tree of life, would be thermophilic, chemosynthetic, and adapted to life in the deep. And here's the thing. When scientists sequence the genomes of modern organisms and build phylogenetic trees, trying to trace back to Luca, the last universal common ancestor, the organism from which all life on Earth descends, the traits they find are consistent with a thermophilic hydrogen metabolizing ancestor. Luca probably lived in a hot environment, probably used hydrogen as an energy source, probably thrived in conditions we'd consider extreme. Luca looks a lot like a vent microbe, which means that when we look at the bacteria and archaea living around hydrothermal vents today, we're not looking at weird offshoots, at life that adapted to an extreme niche. We're looking at something close to the original, the baseline, the blueprint that worked 4 billion years ago and still works now. Photosynthetic life, by comparison, is the evolutionary newcomer, the late arrival that took over the surface because it found an energy source, sunlight, that was abundant and reliable and didn't require living next to a volcanic vent. But it's built on a foundation of chemosynthesis, the enzymes, the metabolic pathways, the fundamental chemistry. All of it traces back to those first microbes, harvesting energy in the dark. We are, all of us, descended from something that lived like this. And if life on Earth began this way, at a vent, in the dark, powered by chemistry alone, then life elsewhere in the universe doesn't need a sun. It needs liquid water, some minerals and heat. That's it. Which means the number of places life could exist just expanded dramatically. Not just planets in the narrow habitable zone of a star, but moons, icy worlds with subsurface oceans, heated by tidal forces, with rocky cores where water and minerals can interact. Places like Europa, like Enceladus, like Titan. Places where there might be vents. And where there are vents, there might be chemistry. And where there's chemistry, there might be life. Not might. There probably is. The giant tube worm has no mouth, no stomach, no anus, no way to ingest food or expel waste. And yet, it's one of the fastest growing invertebrates on Earth, Riftia pachyptila. The name means thick-skinned tube worm, though that hardly captures the strangeness. Picture a white tube, the thickness of your arm, rising 8 feet from the seafloor. The top flares open, revealing a crimson plume that looks like a feathery flower, blood red and delicate, waving slowly in the current. The plume is made of specialized tissues packed with hemoglobin, more hemoglobin per volume than your blood contains, giving it that deep red color. That plume is everything. It's the worm's lifeline. The hemoglobin binds hydrogen, sulfide, and oxygen simultaneously, 2 chemicals that normally react violently with each other. The worm transports both down through its body to a specialized organ called the trophisome, which occupies most of the internal space where a digestive system would normally be. The trophisome is packed with bacteria, billions of them, representing 15 to 25% of the worm's body weight. These bacteria are chemosynthetic, oxidizing the hydrogen sulfide to extract energy, producing organic compounds that the worm absorbs directly into its tissues. No eating, no hunting. The worm is a living greenhouse farming bacteria inside its own body, and it grows fast. According to research published in 2004 by biologist Monica Bright at the University of Vienna, Riftia can grow nearly 3 feet per year under optimal conditions, achieving its full adult size in just a few years. That's extraordinary for a deep-sea organism where most life proceeds slowly, conserving energy in the cold and nutrient-poor environment. But tube worms aren't in a nutrient-poor environment. They're rooted next to a chemical factory, bathed in hydrogen sulfide, surrounded by a constant energy source. They live fast because they can, and they live fast because they have to. Hydrothermal vents aren't permanent. The geology that creates them is dynamic. Seafloor spreading, volcanic activity, tectonic shifts. A vent might flow vigorously for a decade, then slow to a trickle as the magma chamber beneath it cools or shifts. When the chemistry stops, the ecosystem collapses, the bacteria die, the tube worms starve. Everything that depended on the vent disperses or dies. So the organisms that thrive here have evolved for boom and bust cycles, grow fast, reproduce quickly, disperse widely. When a new vent opens up somewhere along the mid-ocean ridge, miles or even hundreds of miles away, the larvae need to find it, settle, and establish a new colony before the window closes. But how do they find it? How does a microscopic larva drifting in the vastness of the deep ocean locate a vent field that might be no larger than a football field? They follow the plume. The mineral-rich water billowing from a vent doesn't just rise straight up. It spreads horizontally, carried by deep ocean currents, creating a chemical trail that can extend for miles. That trail contains sulfur compounds, temperature anomalies, and specific isotopic signatures, and vent larvae, tube worms, mussels, crabs, can detect it. They have chemoreceptors tuned to the exact chemical profile of vent fluid. Once they sense it, they swim or drift up the gradient toward higher concentrations until they reach the source. It's an olfactory treasure map written in sulfur and heat. The Yeti crab, Kiwa hirsuta, discovered in 2005 near Easter Island, takes a different approach. This pale, eyeless crab is covered in dense bristles, especially on its claws, and those bristles are coated with filamentous bacteria. The crab doesn't have the internal symbiotic bacteria that tube worms do. Instead, it gardens them externally. Researchers from the Monterey Bay Aquarium Research Institute observed Kiwa using its claws to wave through the vent fluid, exposing the bacteria to hydrogen sulfide and oxygen. Then it scrapes the bristles across specialized mouthparts, harvesting the bacteria and consuming them. It's actively cultivating its food source, managing a crop, and it's territorial. Kiwa will defend its patch of vent field aggressively, using those bristly claws to fend off rivals. Because access to the vent fluid isn't unlimited, there are optimal zones, close enough to get hydrogen sulfide, far enough to avoid being scalded by the hottest water. The crabs cluster in those zones, competing for real estate the way surface crabs compete for tide pools. Then there are the mussels. Bathymodiolus species colonize vents around the world, forming dense beds around active vents. Like tube worms, they harbour symbiotic bacteria in specialized gill tissues. But some Bathymodiolus species have two types of bacteria living inside them. One that oxidizes sulfur, another that oxidizes methane. They're dual-fuel organisms, able to harvest energy from whichever chemical is more abundant at their particular vent. Research published in 2018 by evolutionary biologist Roxanne Beinart at the University of Rhode Island showed that these mussels can adjust the ratio of their bacterial partners depending on environmental conditions. If sulfur levels drop and methane increases, the methane-oxidizing bacteria multiply. If the chemistry shifts the other way, the sulfur oxidizers take over. The mussel doesn't control this consciously, of course. The bacteria themselves respond to the available nutrients, blooming when conditions favor them. But the result is a symbiotic system that can adapt in real time to a changing chemical environment. This is a form of flexibility most organisms don't have. You can't change your metabolism based on what's for dinner, but if your metabolism is outsourced to bacteria, and those bacteria can shift populations dynamically, you've got options. And then there are the predators. Not everything at a vent is a filter feeder or a bacterial farmer. There are fish, octopuses, crabs that don't cultivate bacteria but hunt other vent organisms. The zorsid fish, elongated, eel-like, patrol the periphery of vent fields, picking off smaller crustaceans. Brasilid shrimps swarm in the plumes, filtering out free-living bacteria. Octopuses den in crevices, hunting at night. But here's what's fascinating. Even the predators are indirectly dependent on chemosynthesis. The food web starts with bacteria. Everything else is eating the bacteria, or eating something that ate the bacteria, or eating something that ate the bacteria. There's no imported energy from the surface. Some organic matter does drift down from above. Marine snow, dead plankton, fecal pellets. But at vent sites, that contribution is negligible. The ecosystem is self-contained, which means if you removed the vents, the entire community would vanish. And some of these organisms have no backup plan. Genetic studies show that many vent-endemic species have extremely low genetic diversity, suggesting they've been isolated for millions of years, evolving in place, never interbreeding with surface populations. They're not just adapted to vent environments, they're obligate vent organisms. They can't survive anywhere else. This is speciation driven by geography and chemistry. Each vent field is an island, separated from other vents by miles of inhospitable seafloor. Larvae can disperse between vents, but the distances are vast, and the currents are unpredictable. Some larvae never find a new vent. They drift until they starve. The ones that succeed found new colonies, but they're genetically isolated from their parent population. Over time, that isolation leads to divergence. A species at one vent field accumulates mutations that a species at another vent field doesn't. Eventually they can't interbreed. They're separate species, even though they descended from a common ancestor just a few million years ago. The deep ocean is full of these isolated lineages. Tube worms on the East Pacific Rise are genetically distinct from tube worms on the Mid-Atlantic Ridge. Yeti crabs near Easter Island are a different species from Yeti crabs in the Indian Ocean. The vents are scattered across the planet, and the organisms living on them are evolving independently, in parallel, under similar selective pressures, but without gene flow between populations. It's like convergent evolution, but within the same lineage. The same basic body plan as tube worm, mussel, crab, but localized adaptations to the specific chemistry, temperature, and predation pressures of each vent field. And the adaptations are extreme. Take Pompeii worms, Alvanella pompejana. These organisms live directly on the sides of black smoker chimneys, in temperatures that fluctuate from near freezing at their head end to over 175 degrees Fahrenheit at their tail end. That's the largest temperature gradient any organism is known to tolerate. The worm's body spans the boundary between two thermal worlds, and it survives by secreting a mucus layer colonized by heat-tolerant bacteria that insulates it from the worst of the heat. Or consider the scaly foot snail, Chrysomelon squamiferum, discovered at vents in the Indian Ocean. Its shell is reinforced with iron sulfide, the same material that forms black smokers. The snail is literally armoured in metal, the only known organism to incorporate iron sulfide into its skeleton. And inside its foot, it has glands packed with symbiotic bacteria, harvesting energy from sulfur compounds. These aren't subtle adaptations. These are radical solutions to radical problems. You can't evolve heat tolerance or metal armour or bacterial farming overnight. These traits took millions of years to develop under intense selective pressure. The organisms that didn't adapt died. The ones that did became something new. And here's the thing. We've only explored a tiny fraction of the ocean floor. Most vent fields remain undiscovered. There are tens of thousands of miles of mid-ocean ridges, subduction zones, back-arc basins where hydrothermal vents could exist. We've visited maybe a few hundred sites in detail with submersibles or ROVs. What else is down there? What other strategies, what other symbioses, what other impossible organisms are thriving in the parts we haven't reached yet? Every time a new vent field is explored, new species are found. Sometimes they're variations on familiar themes, another tube worm, another Yeti crab. But sometimes they're completely unexpected. The scaly foot snail wasn't predicted. The Yeti crab wasn't predicted. They were discovered by accident because someone happened to send a camera or submersible to the right place at the right time. The deep ocean is vast. The vents are rare, scattered, ephemeral, and the life clinging to them has been evolving in isolation, in darkness, under conditions we barely understand for hundreds of millions of years. We're not looking at a complete picture. We're looking at a few pixels in a massive image, and we're trying to infer what the whole thing looks like. But here's what we know. Life at hydrothermal vents is not just surviving, it's thriving, it's diverse, it's innovative. It's found solutions to problems we didn't think had biological answers. And if life can do this here, in scalding water, in crushing darkness, in isolation from the sun, then life can probably do it anywhere. There's a story we've been telling ourselves about how life began. It goes like this. 4 billion years ago, Earth had oceans and a thick atmosphere. Lightning struck the surface, energizing simple molecules, methane, ammonia, water vapor. Those molecules reacted, forming amino acids and nucleotides, the building blocks of proteins and DNA. These building blocks accumulated in tidal pools or shallow seas, a primordial soup of organic compounds. Eventually, through some combination of chance and chemistry, a molecule figured out how to copy itself. That molecule became more complex, membranes formed around it, metabolism emerged, and from that single self-replicating molecule, all life descended. It's a beautiful story, and it might be completely wrong. The primordial soup hypothesis has problems, big ones. For one thing, tidal pools are exposed to ultraviolet radiation. which would destroy complex organic molecules as fast as they formed. Early Earth had no ozone layer. The surface was getting hammered by UV light from the sun and cosmic rays from space. Any delicate chemistry happening in shallow water would have been ripped apart before it could organize into anything resembling life. For another thing, the ocean is huge. If organic molecules did form in the open ocean, they'd be diluted to homeopathic concentrations, drifting apart faster than they could interact. You need concentration. You need molecules colliding frequently enough for reactions to proceed. A dilute soup doesn't give you that. And then there's the energy problem. Building complex molecules from simple ones requires energy input. On the surface, that energy could come from lightning or UV radiation, but those are intermittent, chaotic. You'd get bursts of energy followed by long periods of nothing. Life, even simple life, needs a continuous energy source, something reliable, something that can drive reactions steadily over time. Hydrothermal vents solve all of these problems. First, they're shielded from UV radiation. 2 miles of water is an excellent shield. Any chemistry happening at the seafloor is protected from the destructive radiation that would have sterilized the surface. Second, vents create natural concentration chambers. The porous mineral structures around alkaline vents, those chemical gardens made of calcium carbonate, silica and iron sulfide, are riddled with microscopic compartments. Molecules can accumulate in those compartments, reaching high concentrations, colliding frequently. The pores act like protocells, tiny reaction vessels where chemistry can proceed in an enclosed space. Third, vents provide a continuous energy source, not chaotic bursts like lightning, but a steady gradient. Hot, alkaline, hydrogen-rich fluid on one side of a mineral membrane. Cold, acidic, carbon dioxide-rich ocean water on the other. That gradient, that difference in temperature, pH and chemical composition, can drive reactions. It's a natural battery, and it never runs out as long as the vent is active. This is the hydrothermal origin hypothesis, and it's been gaining momentum for decades. The idea was first proposed seriously in the 1980s by Günther Wechtershäuser, a German chemist and patent lawyer who approached the origin of life from a thermodynamic perspective. Wechtershäuser argued that life didn't start with replication, it started with metabolism, with the ability to harvest energy from the environment and use it to build more complex molecules. Replication came later, once the metabolic machinery was sophisticated enough to copy itself. His model, called the iron-sulfur world hypothesis, focused on the surface of iron and nickel sulfide minerals, the same minerals that form around hydrothermal vents. These minerals, Wechtershäuser argued, could act as catalysts, speeding up reactions that would otherwise proceed too slowly. And the reactions he focused on weren't random, they were reductive, reactions where molecules gain electrons, becoming more reduced, more energy-rich. Why does that matter? Because the molecules of life, amino acids, lipids, sugars, nucleotides, are all reduced compounds. They're rich in carbon-hydrogen bonds. Building them requires taking oxidized molecules like carbon dioxide and adding electrons, adding hydrogen, reducing them. That's hard to do spontaneously. But at a hydrothermal vent with hydrogen gas bubbling up from below and minerals acting as electron donors, reduction happens naturally. Wechtershäuser's ideas were theoretical at first, but over the past 2 decades, experimental work has started to back them up. In 2008, a team led by microbiologist William Martin at the University of Düsseldorf published research showing that the core metabolic pathways shared by all life on Earth, glycolysis, the citric acid cycle, the synthesis of amino acids, could have originated as non-enzymatic chemistry on the surface of iron-sulfur minerals. These pathways don't require DNA or proteins, they're just chemistry, proceeding naturally when the right ingredients are present. In 2019, researchers at University College London, led by chemist Nick Lane, demonstrated that simple organic molecules, fatty acids, amino acids, spontaneously assemble into vesicles under simulated alkaline vent conditions. These vesicles aren't alive, but they're a step in that direction. They're membranes, compartments, boundaries between inside and outside, and they form without any biological intervention. Lane's work focuses specifically on alkaline hydrothermal vents, which are different from the black smokers. Black smokers are hot, acidic, violent. Alkaline vents are cooler, around 100 to 200 degrees Fahrenheit, and they produce fluid that's basic, rich in hydrogen and methane. These vents create mineral structures called white smokers, delicate towers made of calcium carbonate and silica, riddled with tiny pores. Lane argues that these pores are the key. Each pore is a natural electrochemical cell. The walls of the pore are made of iron-nickel sulfide minerals, which are semiconductors. They can transfer electrons. On one side of the wall you have alkaline vent fluid, pH around 9 to 11. On the other side, acidic ocean water, pH around 5 to 6. That pH difference creates a proton gradient, a difference in hydrogen ion concentration, across the mineral membrane. And here's where it gets interesting. All life on Earth uses proton gradients to generate energy. Your cells have mitochondria. Inside the mitochondria, proteins pump protons across a membrane, creating a concentration difference. Then, other proteins, ATP synthase, let the protons flow back, and that flow drives the synthesis of ATP, the energy currency of the cell. Every time you breathe, every time you move, you're using proton gradients to power your metabolism. That's not a coincidence. That's fundamental. Every living thing, from bacteria to blue whales, uses proton gradients, which means the machinery for harnessing proton gradients is ancient, universal. It predates the split between bacteria and archaea, the two deepest branches of the tree of life. Lane's hypothesis is that life didn't invent proton gradients. Life inherited them. The first cells didn't evolve proton-pumping proteins from scratch. They evolved in an environment, alkaline hydrothermal vents, where proton gradients already existed naturally across mineral membranes. The first metabolism was just tapping into the gradient that was already there, using it to drive reactions, to build organic molecules from hydrogen and carbon dioxide. Only later, after millions of years, did cells evolve proteins capable of generating their own proton gradients. At that point they could leave the vents, they could colonize the open ocean, the surface, eventually the land. But the core machinery, the dependence on proton gradients, remained. Because it worked. Because it was baked into the fundamental chemistry of life from the very beginning. If this is true, then the earliest life wasn't free living, it was anchored. Growing in the pores of hydrothermal vent minerals, harvesting energy from hydrogen, using naturally occurring proton gradients to power metabolism, and the chemicals it needed, hydrogen, carbon dioxide, minerals like iron and nickel, were all abundantly available in vent environments. This scenario also explains why Luca, the last universal common ancestor, appears to have been a thermophile. When scientists reconstruct Luca's metabolism by comparing the genes shared by all living organisms, they find evidence of a high-temperature lifestyle. Luca probably lived in water around 175 to 200 degrees Fahrenheit. Luca probably used hydrogen as an energy source. Luca probably thrived in conditions we now find at hydrothermal vents. Luca wasn't at a vent by accident. Luca was there because that's where life began. And if life began at hydrothermal vents on Earth, then it could begin the same way anywhere else. You don't need a planet with a stable atmosphere, a protective ozone layer, a temperate surface environment. You just need an ocean. And below that ocean, a rocky core hot enough to drive hydrothermal circulation and dissolved gases. Hydrogen, carbon dioxide, methane. That's it. That's the minimum requirement. Europa, Jupiter's fourth largest moon, has a liquid ocean beneath an ice shell 10 to 15 miles thick. The ocean is in contact with a rocky mantle. Tidal forces from Jupiter's gravity flex Europa's interior, generating heat. That heat could drive hydrothermal circulation. There could be vents on Europa's ocean floor right now, erupting hydrogen and minerals into the water. And if there are vents, there could be chemistry. And if there's chemistry, there could be life. Enceladus, a moon of Saturn, is smaller but even more tantalizing. In 2005, the Cassini spacecraft observed plumes of water vapor erupting from cracks in Enceladus's south polar region. Those plumes contain water, salt, silica particles, and, critically, molecular hydrogen. The presence of hydrogen suggests hydrothermal activity. Water is reacting with hot rock somewhere beneath the ice. And, when Cassini flew through the plumes, analyzing their composition, it detected organic molecules, carbon-based compounds, Not life, but the ingredients for life. We didn't have to land on Enceladus. We didn't have to drill through the ice. The moon is venting its ocean into space, and we flew through it, sampling the water directly. And we found hydrogen. We found organics. We found evidence of the exact conditions that, on Earth, give rise to life. Titan, Saturn's largest moon, has liquid methane lakes on its surface and possibly a subsurface water ocean. Triton, Neptune's largest moon, shows evidence of cryovolcanism, eruptions of water and ammonia. Pluto, at the edge of the solar system, might have a subsurface ocean kept liquid by radioactive decay in its core. These aren't distant exoplanets light years away. These are worlds in our own solar system, and several of them might have the conditions necessary for life. Not life as we see it on Earth's surface, photosynthetic, oxygen-breathing, sun-dependent. Life as we see it at hydrothermal vents, chemosynthetic, hydrogen-fueled, thriving in darkness. If the origin of life requires vents, and vents are common wherever you have liquid water and a heat source, then life might be common too. Not rare, not a fluke, but an inevitable consequence of planetary chemistry. A phase transition that happens when the right conditions persist long enough. We've been searching for life in the wrong places because we defined life by the wrong parameters. We looked for Earth-like planets in the habitable zones of sun-like stars. But life doesn't need a star. It needs heat, water, and minerals. And those are abundant. The universe might be full of vent worlds, oceans beneath ice, chemistry in the dark, microbes, or their alien equivalents, clustering around hydrothermal plumes, farming hydrogen, evolving in isolation for billions of years. We wouldn't see them with our telescopes. They wouldn't produce biosignatures in their atmospheres. They'd be invisible, hidden, silent. But they'd be alive. And if we want to find them, we don't need to look light years away. We need to go to Europa, to Enceladus, to the ocean worlds in our own backyard, drill through the ice, send a probe into the deep and look for the plumes, look for the vents. Because where there are vents, there might be life. And it might look a lot like home. Evolution doesn't need sunlight. It just needs time, isolation, and pressure. At hydrothermal vents, all three are abundant. Each vent field is an evolutionary island, separated from other vents by miles of barren seafloor, surrounded by water so cold and nutrient-poor that most vent organisms can't survive the crossing. The larvae that do make it, drifting on deep ocean currents for weeks or months, navigating by chemical gradients they can barely detect, arrive at a new vent and colonize it. But they're cut off, genetically isolated, and isolation is the engine of speciation. Over millions of years, populations at different vents diverge, mutations accumulate, selective pressures differ slightly from site to site. One vent runs hotter, another has more methane than sulfide, another sits deeper where pressure is more extreme. The organisms adapt to their local conditions. Eventually they can't interbreed with their cousins at other vents. They're separate species. This process has been playing out for at least 400 million years, possibly much longer. And the result is a global mosaic of vent-endemic species, each lineage shaped by its particular geography and chemistry, each population evolving in parallel but independently. Genetic studies confirm this. In 2001, biologist Robert Vryenhoek at the Monterey Bay Aquarium Research Institute published research comparing tube worm populations from vents along the East Pacific Rise, the Mid-Atlantic Ridge, and back arc basins in the Western Pacific. The populations are morphologically similar. They're all giant tube worms, all hosting symbiotic bacteria, all living the same basic lifestyle. But their DNA tells a different story. The populations have been isolated for millions of years. Gene flow between them is negligible or non-existent. They've diverged to the point where they're classified as separate species, even though they occupy nearly identical ecological niches and look almost indistinguishable to the untrained eye. This is parallel evolution. The same solution to the same problem, arrived at independently, multiple times, across vast distances. And it's everywhere at hydrothermal vents. Yeti crabs, for instance, have evolved at least twice. Kiwa hirsuta, the species discovered near Easter Island, is genetically distinct from Kiwa puravida, found at vents off Costa Rica. Both species are pale, eyeless, covered in bristles colonized by bacteria. Both farm their bacterial partners by waving their claws through vent fluid, but they're separated by thousands of miles and millions of years of evolution. They didn't inherit these traits from a common ancestor that looked like them. They each evolved the same traits independently because those traits work in vent environments. The same pattern appears in mussels, shrimp, snails, fish. Convergent evolution at the morphological level, but genetic isolation at the population level. Each vent field is running its own experiment, testing variations on the same evolutionary themes. And here's where it gets philosophically interesting. If life can evolve the same solutions independently on the same planet, in slightly different locations under slightly different conditions, what does that say about the likelihood of similar solutions arising on completely different planets? If chemosynthesis works at Earth's hydrothermal vents, it should work on Europa's. If symbiosis between animals and bacteria is a successful strategy here, it should be successful there. If organisms evolve metal-reinforced shells or heat shock proteins or bacterial farming in response to vent chemistry on Earth, similar pressures on another world should produce similar innovations. The details would differ. The organisms wouldn't look identical. Evolution is path-dependent. It builds on whatever genetic variation happens to be available at the time. But the underlying principles, the strategies, the biochemical solutions, those might be universal. Because chemistry is universal. Physics is universal. The laws that govern how molecules interact, how energy flows, how systems organize, those don't change from planet to planet. If hydrogen sulfide is a viable energy source for life on Earth, it's a viable energy source for life anywhere. If proton gradients can power metabolism here, they can power metabolism on Enceladus. This is the argument for convergent evolution at the cosmic scale. Not that aliens will look like us, but that alien biochemistry will face the same constraints, solve the same problems, and arrive at similar solutions. Not identical, but recognizable. Analogous. And hydrothermal vents give us a preview of what that might look like. Consider the deep biosphere. We're not just talking about organisms living on the seafloor around vents. We're talking about life inside the rock beneath the seafloor, in the crust itself. In 2013, research led by microbiologist Katrina Edwards at the University of Southern California revealed that the oceanic crust is teeming with microbial life. Bacteria and archaea living in fractures and pores in the basalt, kilometers below the seafloor, sustained by geochemical reactions between rock and seawater. These organisms are slow. They reproduce maybe once every 100 years, maybe once every 1,000. They're not building complex ecosystems or farming bacteria on their claws. They're barely metabolizing. But they're alive, and they've been alive in some cases for millions of years, isolated in tiny pockets of rock, never exposed to sunlight, never interacting with surface life. This is endolithic life, life inside stone, and it fundamentally expands the definition of a habitable environment. You don't need liquid water in contact with vent fluid, you just need water in contact with a rock. Any water, any rock. As long as there's a chemical disequilibrium, something oxidized on one side, something reduced on the other, microbes can extract energy, which means the habitable zone isn't just oceans, it's crusts, subsurface aquifers, fractures in bedrock, anywhere water can seep and chemistry can proceed. On Mars, for instance, the surface is frozen and irradiated, but there's evidence of liquid water trapped in aquifers deep underground, insulated from the cold, possibly in contact with warm rock heated by residual geothermal activity. Could there be microbes down there? Slow metabolizing, ancient, isolated for billions of years? We don't know. But the existence of Earth's deep biosphere suggests it's possible. More than possible. Likely. In 2018, a study published in Proceedings of the National Academy of Sciences estimated the total biomass of Earth's subsurface microbial life. The number is staggering. 15 to 23 billion tons of carbon, representing 10 to 20% of all biomass on the planet. That's more than all the humans, all the livestock, all the wild animals combined. And it's living underground, in rocks, in darkness, sustained by geochemistry. We're the minority. Surface life, photosynthetic life, the entire biosphere we see and interact with. That's the thin film on top. The bulk of Earth's biomass is below, in the crust, living slowly, invisibly, powered by the planet's interior heat. If Earth's biosphere extends kilometers into the crust, why wouldn't Europa's? Why wouldn't Enceladus's? If life can colonize fractured basalt on Earth, it can colonize fractured ice on an ocean world. The chemistry is different, but the principle is the same. Find a gradient, exploit it, persist. And if subsurface life is the norm rather than the exception, then the universe is far more alive than it appears. Not in ways we can easily detect, not producing radio signals or altering planetary atmospheres, but present, quiet, microbial, thriving in the dark. This raises a profound question. What is the default state of life in the universe? We've always assumed Earth is the model, that life, if it exists elsewhere, will be like life here. Diverse, complex, photosynthetic, surface-dwelling. But what if Earth is the anomaly? What if the default is subsurface, chemosynthetic, slow? What if most life in the universe looks like Earth's deep biosphere, sparse, patient, invisible, and the explosion of biodiversity on our surface is the rare event, the fluke, the consequence of photosynthesis unlocking a vast new energy source? In that scenario, Europa isn't a marginal candidate for life. It's the standard model, a world with a rocky core, liquid water, geochemical activity, and microbes living in the crust and around hydrothermal vents. That's the baseline. That's what most life-bearing worlds look like. Earth, with its oxygen atmosphere and forests and coral reefs and vast surface ecosystems, powered by a star, is the outlier, the special case, the exception. This perspective flips astrobiology on its head. We've been searching for Earth analogues, looking for planets in the habitable zone with atmospheres we can analyse for biosignatures. But if most life is subsurface, it won't produce atmospheric biosignatures. It won't be detectable by transit spectroscopy or direct imaging. It'll be hidden, locked away beneath kilometres of ice or rock, accessible only to direct exploration, which means finding extraterrestrial life might require going there, landing, drilling, sending submarines into alien oceans. It's not a telescope problem, it's an engineering problem. And here's the irony. We already have the technology, the same submersibles and ROVs we use to explore Earth's. Hydrothermal vents could, with modifications, explore Europa's. We know how to build pressure vessels that can withstand extreme depths. We know how to navigate in complete darkness using sonar and chemical sensors. We know how to collect samples and analyse them for signs of life. The challenge isn't the science, it's the distance and the ice. Europa's ice shell is 10 to 15 miles thick. Drilling through it would be a monumental engineering effort, requiring either a heated probe that melts its way down over weeks or months, or a more aggressive drilling system powered by a nuclear reactor. Both concepts have been studied. Both are theoretically feasible, but neither has been tested in the field, and the stakes are high. You get one shot. If the probe gets stuck halfway down, if the ice refreezes behind it, if it loses power before reaching the ocean, mission over. But if it works, if a probe makes it through the ice, deploys a submarine, descends into Europa's ocean, and finds a hydrothermal vent, if it finds tube worms, or their alien equivalents, clustering around a smoker, farming bacteria, thriving in the dark, that would be the most important discovery in human history. Proof that life is not unique to Earth. That the chemistry we see at our vents is universal. That wherever there's water, heat, and minerals, life finds a way. And it wouldn't just be microbial. The existence of complex vent ecosystems on Earth, animals with sophisticated symbioses, predators and prey, entire food webs, suggests that chemosynthetic ecosystems can support complexity. You don't need sunlight to build a biosphere, you just need energy and time. So Europa might not just have microbes, it might have its own version of tube worms, its own yeti crabs, maybe even something more complex, something that moves, something that hunts. We don't know. We can't know until we go there. But the fact that life on Earth evolved complex, thriving ecosystems in complete darkness, in isolation from the sun, suggests that Europa could too, that Enceladus could, that any world with a subsurface ocean and geothermal activity could host not just microbial mats, but animals, alien animals, chemosynthetic, vent-dependent, adapted to crushing pressure and eternal night. They wouldn't look like anything we've seen, but they'd be alive, and they'd prove that life, real, complex, thriving life, doesn't need a star. It just needs a vent. We've mapped the surface of Mars in higher resolution than we've mapped the ocean floor. Think about that. We've sent rovers to another planet, analysed its rocks, searched for biosignatures in ancient riverbeds. But here, on Earth, more than 80% of the ocean remains unexplored. We have better topographic maps of Venus, hidden beneath a crushing sulfuric acid atmosphere, than we do of the Mariana Trench, and hydrothermal vents. We've found maybe a few hundred sites, but the global mid-ocean ridge system spans 40,000 miles, the longest mountain chain on Earth, and most of it has never been surveyed with the resolution needed to detect vents. There could be thousands of active vent fields we haven't discovered, tens of thousands. Each one is a potential hotspot of biodiversity, a reservoir of species found nowhere else, a laboratory where evolution has been running experiments in isolation for millions of years. What's down there in the parts we haven't seen? In 2020, researchers exploring the Mid-Cayman Rise, a vent field in the Caribbean, discovered the world's deepest known hydrothermal vents, nearly 3 miles down. The site, called BB Vent Field, sits at the bottom of a tectonic rift where the ocean floor is literally splitting apart. And the organisms they found there were unlike anything seen before. New species of shrimp, anemones with translucent bodies glowing faintly in the lights of the ROV, microbial mats with metabolic pathways that don't fit existing models. This was 2020, not the 1970s, not the early days of deep-sea exploration. We're still finding fundamentally new things every time we look, and we're barely looking. The funding for deep-sea research is a rounding error compared to space exploration. NASA's budget for 2024 was over $25 billion. NOAA's Ocean Exploration Program, around $50 million. That's 0.2%. We spend more on a single Mars mission than we do on exploring Earth's oceans in a decade. Why? Partly because space is visible, aspirational. You can point a telescope at Mars and see it. You can show people pictures of rovers on red dust plains. The ocean is opaque, hidden, out of sight, out of mind. But also because exploring the deep ocean is hard, harder in some ways than exploring space. The pressure at vent depths is extreme, over 3,000 pounds per square inch, enough to crush most materials. Seawater is corrosive, darkness is total, communication is limited, radio waves don't penetrate water, so you're reliant on acoustic signals or tethered cables. Every mission requires a research vessel, a submersible or ROV, a trained crew, and days or weeks at sea. It's expensive, it's slow, and the public doesn't pay much attention. But the discoveries waiting in the deep might be more important than anything we'll find on Mars. Consider extremophiles, organisms adapted to extreme conditions. Hydrothermal vent bacteria can survive temperatures that would denature most proteins, pH levels that would dissolve tissue, pressures that would collapse cells. These organisms have enzymes that function in boiling water, membranes that remain stable under crushing force, DNA repair mechanisms that fix radiation damage faster than it accumulates, and we're just beginning to understand how they do it. In 2019, a team at the University of California, Berkeley, isolated a strain of bacteria from a vent field that produces a heat-stable enzyme capable of breaking down plastics. The enzyme, called PETase, can degrade polyethylene, terephthalate, the plastic used in bottles and packaging, into its constituent molecules which can then be recycled. At room temperature, this process takes years. At the elevated temperatures the enzyme evolved for, it takes hours. This isn't theoretical. Companies are already developing industrial processes based on this enzyme, aiming to create closed-loop recycling systems for plastics that currently end up in landfills or oceans. That discovery came from a single microbe, from one vent field. There are thousands of vent fields, millions of microbial species, most of them uncharacterized, many of them producing novel enzymes, metabolic pathways, chemical compounds we've never seen before. Pharmaceutical companies are paying attention. Extremophils produce bioactive compounds, antibiotics, antivirals, anti-cancer agents, because they're constantly competing with other microbes in hostile environments. The chemical arms race at a hydrothermal vent where bacteria fight for space and nutrients using toxins and signaling molecules, has been going on for hundreds of millions of years. The compounds that work, that give an organism an edge, are refined by natural selection to be potent, specific, and stable under extreme conditions. In 2015, a deep-sea bacterium yielded a compound called leguminomycin, which showed promising activity against drug-resistant tuberculosis. In 2022, another vent microbe produced an enzyme that could degrade PFAS, forever chemicals that persist in the environment and accumulate in living tissue. These discoveries are accidental, byproducts of basic research. Scientists studying vent ecosystems for their own sake, trying to understand how life works in extreme environments and stumbling onto applications with massive commercial and environmental value. But we're not systematically searching. We're sampling opportunistically, collecting a few microbes here and there, culturing the ones that grow easily in the lab, and hoping we get lucky. Imagine what we'd find if we actually tried. If we surveyed every vent field, catalogued every species, sequenced every genome, characterized every enzyme. The biotechnology locked away in those organisms could revolutionize medicine, materials science, environmental remediation. And that's just Earth. If we find life on Europa or Enceladus, life that evolved independently using different biochemistry, adapted to different conditions, the potential applications are unimaginable. Alien enzymes, alien metabolic pathways, chemistry we've never conceived of because it didn't evolve here. But let's bring it back to the immediate question. What don't we know about Earth's hydrothermal vents? A lot. We don't know how many vent fields exist. We don't know how long an average vent remains active, or how organisms disperse when a vent shuts down. We don't fully understand the symbioses between animals and bacteria, how the partnerships form, how the partners communicate, how the host controls bacterial populations. We don't know if vent ecosystems are self-sustaining over geological timescales, or if they depend on occasional immigration from surface populations. We don't know if the genetic isolation of vent-endemic species makes them more or less resilient to environmental change. We don't know how climate change, warming oceans, acidification, changes in deep currents, will affect vent communities. And we definitely don't know what's living in the unexplored regions, the Southern Ocean, the Arctic, the Hadal Zone, the deepest trenches below 20,000 feet, where pressure exceeds 16,000 psi. There are vents down there. We've detected them with sonar, seen the plumes, but we've never sent a submersible deep enough to observe them up close. What's adapted to those conditions? What metabolic strategies? What body plans? What evolutionary innovations exist in environments so extreme we can barely build instruments to survive them? In 2012, filmmaker James Cameron descended to the bottom of the Challenger Deep, the deepest point in the Mariana Trench, nearly 7 miles down. He spent 3 hours on the seafloor collecting samples, filming, observing, and he saw life. Amphipods, microbial mats, organisms thriving at pressures that would compress steel, but he didn't see a vent. The Challenger Deep might not have active vents, or they might be elsewhere in the trench, in areas he didn't visit. We don't know. The trench is too big, too deep, too difficult to explore comprehensively. And the Challenger Deep is just one trench. There are dozens of others scattered across the Pacific, the Atlantic, the Indian Ocean, each one potentially harbouring its own ecosystems, its own endemic species, its own evolutionary experiments. We are infants in the deep. We've taken our first steps, glimpsed a few wonders, and declared ourselves knowledgeable. But the ocean is vast, and we've seen almost none of it. There's a philosophical implication here too. If Earth, our home planet, the world we've inhabited for 300,000 years, still holds entire ecosystems we didn't know existed until 50 years ago, what does that say about the limits of our knowledge? We thought we understood the requirements for life. We thought we knew where to look, what to expect, and we were wrong, completely wrong. The discovery of hydrothermal vent ecosystems wasn't an incremental addition to existing knowledge. It was a paradigm shift, a revelation that life is more flexible, more creative, more resilient than we imagined. And if we were that wrong about our own planet, how wrong might we be about the universe? We've spent decades searching for extraterrestrial intelligence, listening for radio signals, assuming that advanced civilizations would build transmitters and broadcast their presence. But what if most life in the universe is subsurface? Silent, invisible to our telescopes because it never evolved technology, never needed to because it's thriving in stable, isolated environments where there's no evolutionary pressure to develop language or tools or curiosity about the stars? What if the universe is teeming with life, but it's all below the surface, in oceans and crusts, chemosynthetic and microbial, living slowly, reproducing rarely, completely unaware of anything beyond its immediate chemical environment? We wouldn't find it with SETI. We'd find it by going there, by drilling through ice, descending into alien oceans and looking at the rocks, the water, the vents. And maybe that's the lesson, that life doesn't announce itself. It hides. It persists quietly in the corners where chemistry allows it, where energy flows and gradients exist, where the universe is out of equilibrium just enough for complexity to emerge. The search for life isn't about listening, it's about looking, closely, in the places we've overlooked because they seemed too hostile, too dark, too alien. But those places, the deep trenches, the subsurface aquifers, the ocean worlds of the outer solar system, aren't alien. They're home to something. Maybe not something we'd recognize at first glance. Maybe not something that thinks or communicates or builds, but something alive. And the fact that we found it here, on Earth, thriving in conditions we thought were uninhabitable, tells us that the universe is far more welcoming to life than we assumed. That the boundaries we drew around the habitable zone were arbitrary. That life doesn't need our permission or our understanding, it just needs chemistry. And chemistry is everywhere. So when we finally send a probe to Europa, when it drills through the ice and releases a submarine into that alien ocean, when it descends through the darkness toward the rocky floor below, what will it find? Maybe nothing. Maybe the ocean is sterile, the chemistry insufficient, the conditions just slightly wrong in some way we didn't predict. Or maybe it'll find a vent, a plume of superheated water rising from a crack in the rock, minerals precipitating out, forming chimneys, and clustered around those chimneys something moving, something feeding, something alive. Not like us, not even like the organisms at Earth's vents, but recognizably, undeniably alive, harvesting energy from hydrogen, building tissues from carbon dioxide, hosting symbiotic microbes in specialized organs, living in the dark because it never needed light, and in that moment, we'll know, life isn't rare. It's not confined to one planet, one chemistry, one path. It's a pattern, a process, a consequence of thermodynamics and time. Wherever water meets rock and energy flows, life finds a way. We've been looking up, asking if we're alone, but the answer was always below. 2 miles down, in the dark, in the heat, in the impossible ecosystems we never predicted. We're not alone. We never were. We just weren't looking in the right place. Until 1977, when we finally descended into the abyss, turned on the lights, and saw the truth. Life doesn't require the sun. It requires chemistry, and the universe is full of it. The alien life we've been searching for isn't light years away. It's here. It's been here for billions of years, thriving, evolving, waiting for us to notice. And now that we have, the question isn't whether life exists beyond Earth. The question is, how common is it? How diverse? How strange? And the only way to find out is to keep descending, keep exploring, keep looking in the places that seem impossible. Because impossible, it turns out, is exactly where life likes to be.